In this section, we'll consider a central example of top-down influence on bottom-up processing, namely volitional attention, also known as endogenous attention. In the last section, I mentioned that one key way that top-down modulation of bottom-up processing happens at a neuronal level is via the release of neuromodulating neurotransmitters into the synaptic clefts of neural circuits that process bottom-up inputs. One example of such a modulatory neurotransmitter is acetylcholine. There are receptors that lie slightly away from the synapse that allow for the regulation of intracellular potential via changes in ligand and voltage-gated receptor conductances. This just means that the likelihood that receptors will pass ions can change as a result of neuromodulation by acetylcholine and other neuromodulators. For example, one of acetylcholine's effects might involve altering the probabilities that NMDA receptors will open. Extrasynaptic muscarinic acetylcholine receptors are of this class and appear to play an important role in network changes that occur with changes in arousal and attention. Cholinergic neurons originating in the nucleus basalis of the basal forebrain, shown here, project widely across cortex. Attention may have its effects on local neural circuitry in part via the release of acetylcholine from cholinergic neurons originating in the basal forebrain nucleus that binds with muscarinic and nicotinic acetylcholine receptors. When such binding occurs, some neurons may become more excitable while others might become less so. If muscarinic acetylcholine receptors lie on inhibitory interneurons, acetylcholine would increase the drive of the inhibitory network that surrounds, synchronizes, and regulates pyramidal cells. This might in turn alter the degree of synchrony or the probability of burst firing. Since neurons are ultimately spike coincidence detectors, changing synchrony or burstiness or rates of firing through such a top-down mechanism would alter information processing in a neural circuit very fundamentally. There are multiple acetylcholine-mediated effects, many more than we can get into here, but a result of cholinergic projections from the forebrain is that attention enhances the likelihood of signal detection and transmission. Attention is not only realized in acetylcholine release in a top-down manner from the basal forebrain to a relatively early visual processing area such as V4, shown here. There's evidence that basal forebrain non-cholinergic neuronal bursting may realize top-down attentional modulation of pyramidal cells' behavior. In particular, it may switch pyramidal cell networks from a tonic or relatively non-bursty to a phasic or relatively bursty mode, perhaps via basal forebrain neurons' GABAergic inhibition of local inhibitory interneurons. Another possibility is that top-down attention alters the dynamics of the local inhibitory network so that it becomes more synchronized, which would increase gamma oscillations in that circuit even before the arrival of bottom-up input, changing the way that the neural circuitry will respond to bottom-up input when it later arrives. If this view is correct, then top-down attentional alteration of bottom-up processing may be realized in the transitioning of the target cortical circuit from a tonic into a bursting mode mediated by NMDA receptor opening with concomitant increases in firing rate. Attentional binding would not be the gluing of features together at a location, but would instead afford complete information processing at an attended location from visual input areas all the way down to the hippocampus. The process might work something like this short animation. Neurons in a visual processing area such as V4 that received top-down cholinergic and non-cholinergic neurotransmitter release, ultimately from the basal nucleus of the forebrain, might change their sensitivity to subsequent spike inputs, altering in turn their spiking pattern and rate, which would then alter downstream neurons' behavior. One metaphor is that attention might set up something like a bucket brigade of processing from early visual areas all the way down to the hippocampus for enhanced identification, but only at the attended location. A lot of difficult neuroscience has yet to be done to fully work out how attention and the changes that it induces in consciousness is realized in top-down control of bottom-up processing via cholinergic and non-cholinergic inputs. And many questions remain to be answered. For example, what circuitry controls the basal forebrain to release neurotransmitters where it does? Be that as it may, we do not have to have a complete understanding of the brain to appreciate that there can be top-down causation in the brain effectively realized by changing the synaptic weights of downstream neurons by altering, for example, the conductances of receptors. This will in turn alter information processing in those downstream neurons. Thus, the biggest point, that patterns in energy can become causal, is rooted in the tiniest point, that conductances of receptors can be altered rapidly and dynamically in light of information processing needs of the whole organism. Ultimately, patterns among energy become causal in the universe only when pattern detectors exist. In us, these have evolved in fascinating ways. In this course, we've considered two such pattern detectors. One, at a cellular level, is a neuron, which is sensitive to the pattern of coincidence or coincident arrival of action potentials. In this temporal pattern, many other patterns can be encoded, such as the spatial pattern of a simple cell tuned to an oriented bar of light, or a neuron tuned to the spatial pattern of the constellation Orion, or really any other pattern at all. Then, at a much tinier level, at the level of receptors, we also considered the pattern detector realized in an NMDA receptor, which is also a coincidence detector, namely of three facts being true: glutamate binding, glycine binding, and then also postsynaptic neuron depolarization. When NMDA receptors open, they effectively change the synaptic weights linking a pre- and postsynaptic neuron, changing both the firing rate and burstiness of postsynaptic neurons, but also altering its responsiveness to subsequent presynaptic inputs. This mechanism of dynamically altering synaptic weights is subject to top-down volitional control via the intermediation of acetylcholine and other neurotransmitters. Amazingly, microscopic NMDA receptors may play at least four important roles in the neuronal basis of mental causation. First, they introduce randomness to the macroscopic domain in the form of spike timing uncertainty, which may then meet or not meet present neuronally realized informational criteria. Second, they may afford the generation of many possible solutions that can meet any set of criteria. Third, they may realize rapid synaptic resetting that permits neurons to escape the impossibility of self-causation by changing the criteria for firing of neurons on the next information processing cycle. And fourth, they transition neural network behavior into a burstier mode of information processing. Some scientists think that this may permit the transfer of bursts as informational packets across a network. Top-down cholinergic, GABAergic, and other neuromodulator input can alter neural function, ultimately at the level of receptors, which is then magnified up to a level of depolarization, which then affects spiking, which in turn alters the rest of the brain. If you wish to learn more about the details of receptor function, you can look at the neural basis of free will, which goes into much greater detail. But suffice it to say that top-down criterial causation can operate on the millisecond timescale of thoughts by rapidly resetting synaptic weights and altering other neural response properties. Volition is closely tied to endogenous attention, which is in part realized in alterations of neurons receiving top-down cholinergic and non-cholinergic inputs from the basal forebrain and other areas, such as the posterior parietal lobe. In this course, I've argued that causation was initially conceived to be a transfer of energy. Later notions of causation emphasized how energy is transformed. Criterial causation emphasizes not how energy is transferred or transformed into a different state, but how patterns of energy, such as coincidence, are transmitted and transformed. In this section, we've considered how a top-down process, such as what we choose to attend to based upon considerations in working memory, can come to alter the dynamics of neural circuitry, ultimately by altering the properties of synapses. Such top-down causation is made possible by altering the behavior of coincidence detectors operating at the molecular level, such as NMDA receptors. For me, the beauty of what evolution came up with is breathtaking, because patterns can be created and destroyed. Unlike the physical substrate in which they are realized, patterns are not subject to the same physical laws that constrain amounts of energy. This permits the emergence of higher-level types of causation of pattern on pattern, or a type of causation that operates over a succession of patterns. This affords a downward and non-epiphenomenal causation where mental events, realized in some neurons' criteria for firing having been met, only occur if elementary particles realize a particular subset of the possible paths open to them. This subset is the subset that embodies not only a physical causal chain of events, but also an informational causal chain of events. Other possible paths that are open to elementary particles which do not also realize an informational causal chain are in essence filtered out from potentially happening at the level of neurons because those paths fail to meet physical or informational criteria for the release of an action potential. All informational causal chains are also physical causal chains, but the vast majority of physical causal chains in the universe are not informational causal chains. What makes some physical causal chains also informational causal chains is that the physically realized criteria for some physical event happening, like the firing of a neuron, are only met when certain informational criteria are also met. If inputs to this neuron are such that action potentials sufficient to make it fire only arrive from presynaptic neurons when some informational conditions are met, then this neuron realizes informational criteria by virtue of imposing physical criteria for firing upon its physical inputs. This information is used by a physical organism to do useful things like find food, mates, and avoid dangers.